Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 23rd video of the chapter 2 of immunology. And in this particular video, I want to focus on the eosinophils. These eosinophils, they belong to a specialized class of the white blood cells, which are known as the granulocytes, and they are known as the granulocytes because they contain granules in their cytoplasm. These eosinophils, they are very important in the parasitic infection, and we will see an example of this in this particular video. And they are also very important in the allergic reactions. If you talk about their origin and development, they are derived from the bone marrow. So the bone marrow is going to give rise to the hematopoietic stem cells. The hematopoietic stem cell is going to give rise to the myeloid progenitor cells, and from these myeloid progenitor cells, all of the granulocytes, they are derived, including the eosinophils, the basophils, and the neutrophils. So the eosinophils, they are actually derived from the bone marrow, particularly from the myeloid progenitor cells. If you talk about their lifespan is very short, and that is about 8 to 18 hours in the blood. Morphologically, these eosinophils, they usually have a size of 12 to 17 micrometer in diameter. They are usually round in shape, and they have got a bilobed nucleus, as you can see in this particular image. If you want to stain them, so usually the eosin or the other acidic dyes, they are used for the staining of the eosinophils, and these eosin or the other acid dyes, they are going to stain the granules of the eosinophils red, making them distinguishable from the other blood cells, particularly from the other granulocytes like the neutrophils and the basophils. So you can see a very clear-cut differences between the eosinophils and the other granulocytes in this particular image. Now, as I've told you that these eosinophils, they are granulocytes. That means they are going to have granules in their cytoplasm. So we are going to talk about the primary or the azerophilic granules of the eosinophils in this particular video. Now, when there is infection of a parasite in the tissue, for example, if this is a tissue and it get infected by a parasite, so what happens is that the eosinophils, they are going to move towards the site of the infection. Now, how this is achieved, how the eosinophils know where to move. So there is a particular process which is known as the chemotaxis and the recognition, and different chemotactic factors like the eotoxin, the interleukin 15, or the other signaling molecules, they are actually directing the eosinophils to move toward the site of the parasitic infections. Now, what the eosinophils, they are going to do then when they reach to the site of the infection? So if this is this, you consider this effector cell as your eosinophils. So if you see over here, the eosinophil, they have got these specialized receptors on their surface, which are known as the Fc receptors. And these Fc receptors are very good at recognizing the parasites. So what happens is that when the tissue that is infected by the parasite, so the antibodies, they are going to get attached to this parasite. So this parasite that has been attached by this antibody, and the eosinophils, they are going to use their Fc receptor to recognize this antibody bound parasite. So once the eosinophil, they recognize the the antibody bound parasite using their Fc receptors, that actually leads to the engulfment through the process of the phagocytosis. Now, once the eosinophils, they have phagocytosed the parasite, so what is going to happen next? Now, this is the granules present in the eosinophils. You've got these granular components over here. So when the eosinophil, they get activated, so say for example by cytokines or the immune complexes like the antibody bound to the antigen, that actually leads to the degranulation of these granules. Now, these granules, they contain a lot of components, and in this particular video, I'm only going to focus on a single one, which is known as the acid phosphatase. Now, keep in mind one thing, that this particular enzyme is a phosphatase. That would mean that when it interacts with its substrate, that is going to remove phosphate groups from there. So this is a phosphatase, and phosphatases are responsible for removing a phosphate group from their substrate protein. Now, for the activation of the acid phosphatase, you need an acidic environment like a pH of 4.5 to 5. Now, if you talk about the phagocytosis, so if this is the parasite that has been phagocytosed by the eosinophil, so this particular structure, the membrane structure, the part of the membrane over here containing the parasite, this particular structure is known as the phagosome. So the phagosome, that would be the parasite plus part of the membrane that has been used for the process of the phagocytosis. Now, when this phagosome that has been formed, the phagosome is going to combine with the lysosome, and the lysosome plus the phagosome, this particular structure is known as the phagolysosome. Now, the acid phosphatase, it actually moves after the degranulation. It interacts with the phagolysosome. The phagolysosome is going to provide an acidic environment to the acid phosphatase, like from 4.5 to 5, and that is going to lead to the activation of the acid phosphatase. And the acid phosphatase, when it gets activated, it is going for the dephosphorylation of a variety of the substrates, for example, the phospholipids in the membrane. And when the phospholipids in the membrane, they get dephosphorylated, that is going to lead to the destruction of the parasite. Now, the acid phosphatase, along with the other enzymes like the proteases, the lipases, and the nucleases, that actually leads to the destruction of the parasites. Secondly, there is also production of the reactive oxygen species, and this reactive oxygen species produced by the eosinophils, they damage the parasite cellular components, aiding in its degradation. So this is how the acid phosphatase that is going to lead to the destruction of the parasite. We will have a detailed discussion on these components in our coming videos. Now, once the parasite that has been converted into pieces, so there comes the process of the antigen presentation. So the degraded parasite peptides, they are then presented on the eosinophil surface via the MHC class 2 molecules, facilitating the adaptive immune response and the clearance of the pathogen from the body. So we will continue the discussion in the next video on the eosinophils.